Amino acids are essential molecules. They are the building blocks of proteins. Proteins are crucial for life, but amino acids have a secret. They possess fascinating optical properties. These properties tell us a lot about their structure and function. Amino acids interact with light in a unique way. This interaction is called optical activity. It helps us understand the three-dimensional arrangement of atoms within these molecules. This knowledge is essential in fields like medicine, biochemistry, and pharmaceuticals. Join me on a journey into the microscopic world of amino acids. We'll uncover the secrets of their optical properties. We'll see how these properties impact our understanding of life itself. When a beam of plain polarized light is passed through an amino acid solution, something amazing happens. The plane of polarized light rotates. This is optical activity. Most amino acids have the ability to rotate plain polarized light. This ability makes them optically active. Glycine is the exception. It does not rotate plain polarized light. It is not optically active. The unique structure of each amino acid dictates whether it possesses this special property. Chirality is a key concept. It's like looking in a mirror. Your left hand is a mirror image of your right hand. They are not superimposable. Amino acids are chiral molecules. They have a central carbon atom bonded to 4 different groups. This carbon atom is called the chiral carbon. It's like the center of the mirror. This chiral carbon gives amino acids their handedness. Like your hands, 2 mirror images of an amino acid exist. They are called enantiomers. This chirality is what makes amino acids optically active. Amino acids can rotate plane polarized light. Some amino acids rotate plane of light to the right, clockwise. These are called dextrorotatory. Other amino acids rotate plane of light to the left, counterclockwise. These are called levorotatory. The direction of rotation is determined by the spatial arrangement of the 4 groups around the chiral carbon. This ability to rotate light in specific directions has important implications. It affects how amino acids interact with other chiral molecules. This interaction is important for biological processes. D and L, a system of labels. Scientists use a special system to label enantiomers. It's called the D and L system. This system is based on the configuration of glyceraldehyde. Glyceraldehyde is a simple sugar. If an amino acid has a similar configuration to D-glyceraldehyde, it's a D-amino acid. If it's similar to L-glyceraldehyde, it's an L-amino acid. This labeling system helps scientists distinguish between the 2 enantiomers. It's important for understanding the biological activity of amino acids. Measuring the rotation of plane of light. How do we measure optical activity? We use an instrument called a polarimeter. It measures the angle of rotation of plane polarized light. A polarimeter has a light source, a polarizer, a sample tube, and an analyzer. The polarizer creates plane polarized light. The sample tube holds the amino acid solution. The analyzer measures the angle of rotation. By analyzing the light that passes through the sample, we can determine the direction and magnitude of rotation. This information helps us identify and characterize amino acids. How much does it rotate? Optical rotation is the observed rotation of plane polarized light. It's measured in degrees. The degree of rotation depends on several factors: the nature of the amino acid, its concentration, the temperature, and the wavelength of light used. Optical rotation is a valuable tool. It helps us determine the purity of a sample. It can also be used to study the kinetics of chemical reactions. By measuring the optical rotation over time, we can track changes in the concentration of optically active compounds. This information provides insights into the mechanisms of reactions. Section 8. Factors that affect the twist influencing optical rotation. Several factors influence the degree of optical rotation. Concentration plays a role. The more concentrated a solution, the greater the rotation. Temperature also matters. Higher temperatures can increase or decrease rotation depending on the substance. The wavelength of light used is crucial. Different wavelengths can result in different degrees of rotation. The solvent used to dissolve the amino acid can also affect the rotation. Finally, the length of the light path through the solution influences the observed rotation. Understanding these factors is essential for accurately interpreting optical rotation measurements. It allows scientists to control experimental conditions. A standard measure, specific rotation. Specific rotation is a standardized measure of optical activity. It takes into account the concentration of the sample, the length of the light path, and the temperature. By using specific rotation, scientists can compare the optical activity of different compounds. It provides a consistent and reliable measure of this important property. Specific rotation is expressed in degrees per unit concentration and path length. It is a fundamental property of optically active compounds. Thanks for watching Life Science Insights. For more such informative content, subscribe to our channel and hit the notification bell. We'll see you in the next video.